So we previously looked at data set number 2. This time we're going to look at data set number 5. This one is one that shows us more of an illustration versus a graph or data table that helps us become more familiar with cells. Recall, we're going to be looking at text and images, and ultimately what we're trying to do is figure out how does this help us understand cells a little bit more deeply than we did prior. So what does this share with us regarding cells? This specific data set contains a ton of vocab, and when you come across things like this that has a lot of words that might be misleading, it is highly important that you annotate the text to make good understanding of what it is that you're actually reading. So let's start by going through the reading. Carcinogenesis, man, they start with a word right there. Carcinogenesis, also called oncogenesis. So if it's also called that, then these two things, they are the same. So carcinogenesis or oncogenesis is the formation of cancer. So these two things, they are the formation of cancer. That would be our definition of those two terms. So oncogenesis, formation of cancer. Cancer is when normal cells are transformed meaning kind of like to change. So when they are changed into cancer cells. So normal cells, they get changed into cancer cells. This process is characterized by changes at the cellular and genetic levels and abnormal cell division. Okay, let's talk about this. Cellular and genetic levels. Whenever we hear the term genetic, we should be thinking of DNA. DNA controls everything about us, and DNA is located inside of cells. So if DNA changes, then cells change. And when cells change, they can, but not always, but they can become damaged. Kind of like what we saw with the picture in our first handout that we received. We had normal cell division and cancer cell division, and we saw that in normal cell division, damaged cells, they go through apoptosis and they die. Damaged cells in cancerous cell division, they become damaged, and those cells, they do not die. They go through abnormal cell division, they continue to grow instead of die. Cell division, it is a process, or it's something that happens, and it occurs or happens in almost all tissues under a variety of circumstances. So pretty much all cells in our body, they will go through cell division like we saw on the previous handout. They go through normal cell division. You have one cell, and it produces two cells that are identical to the first. That's normal cell division. What would cause some of those things to happen? Well, that could depend. Why would this cell create two new cells? We'll get more into why that would happen later on. But do understand that this happens in all tissues, except for really our nervous tissue, which is kind of why if you get a spinal injury and you injure your spinal cord, you are paralyzed, and that tends to be lifelong because those cells cannot make new cells. Normally, the balance between proliferation, formation. Proliferation is a fancy word for formation meaning to make or build something. So normally the balance between making cells and apoptosis, which we already have identified as cell death, It's maintained to ensure the integrity of tissues and organs. Integrity of tissues and organs, this means to kind of have things work properly. If the cells of our body have good integrity, then our cells are working and functioning properly. Our heart is beating as it normally should. The cells in the heart, which is an organ, are reproducing as they normally should, so on and so forth. According to the prevailing accepted theory of carcinogenesis, we saw that up here, so carcinogenesis, so we know that is the formation of cancer. The somatic mutation theory, which is mutations in DNA, lead to cancer. So somatic mutation theory, they are this. That is the definition of the somatic mutation theory. It's when changes in DNA lead to cancer. They disrupt or change, another word for change, the normal processes by disrupting the genes that control cell division. So we have genes. Genes, they are part of our DNA. They are actually on our DNA. Our DNA has a lot of genes on it. We have genes that control eye color, genes that control how tall we're going to get, genes that determine whether we will or will not get certain forms of cancer. If something is genetic, it is controlled by genes on our DNA. So when we disrupt or change things that affect genes, specifically genes that control when a cell dies, apoptosis, and when cells begin to split in half, we do upset or change the balance between proliferation and cell death. When we change that balance between proliferation and cell death, we get uncontrolled cell division, and we end up with a lot of damaged cells multiplying very fast like we saw previously. Only certain mutations lead to cancer. Not all mutations lead to cancer. And it says that right here. Most mutations do not. All right, that's a lot of information. Let's look at our graphic. Maybe that can simplify things a little bit. The first step, we have one cell, and we have a mutation. Mutation. It is a change in DNA. So when there is a change in DNA, we inactivate, meaning turn off, tumor suppressor genes. Tumor suppressor genes, they control when a cell will divide, meaning when a cell will go from one cell and make two cells. That is controlled by a tumor suppressor gene. And when cells, or they, will stop dividing. So if there is a mutation, if there is a change in DNA that turns off this gene which controls when cells will divide and when they will stop dividing, we end up with multiple cells forming pretty quickly. We see proliferation. Once again, we identified proliferation over in our reading as the formation of cells. At this point, these cells are not cancerous. They just have a section of DNA where this gene, it no longer works. Other than that, the cells are normal, but they just kind of go through growth much quicker, and this would maybe result in bumps or kind of pre-tumorous buildups of tissue, or maybe lumps on the skin, for example. Additional mutations or changes in DNA can happen to these cells. If they happen to these cells and they happen to inactivate or turn off a DNA repair gene, then we have more problems. This will not always happen to these cells. We're saying when changes in DNA do turn off DNA repair genes, then we can have more problems. That is why this is said to be pre-cancer, but it's not cancer because it's still kind of functioning normally, other than going through mitosis and making new cells really, really rapidly. What DNA repair genes do is they proofread DNA. Think of proofreading when you maybe proofread in some of your writing classes where you have to read what you have written and check it for errors. That's what DNA repair genes do. Do understand that DNA, it's found in cells, and do understand that these two cells are kind of born from this original cell. These two cells, they are identical to this cell. So these two cells, they have the exact same DNA as this cell. When we are making two cells, this cell has one set of DNA. This cell has one set of DNA, this cell has one set of DNA. In order for this guy to create these two, it has to make a copy of DNA. And we're saying that DNA repair genes, they proofread the DNA after it's been copied, but before these two cells are born. So this cell will make a copy of its DNA. DNA repair genes, they proofread it, and once they say, yeah, everything's good, it's passed on to these two new cells, and those cells are born. So, if a cell has experienced a change in the DNA that turns off tumor suppressor genes, and then experienced second mutations or changes in DNA that turn off DNA repair genes, then we have changes in proto-oncogenes forming what are known as oncogenes. A proto-oncogene is a gene or a section of DNA that helps cells grow. Just like else, cells have a lifespan, they are born small and they get larger in size. So these genes, the mutation, they are turned off. They no longer function. When these cells are turned off, they form oncogenes, which are now permanently turned on. And an oncogene is a proto-oncogene that has just been modified. It has been changed. So these genes, they help cells grow and they are turned off typically. But once this has been changed, the proto-oncogene becomes an oncogene and is permanently turned on, meaning that the genes that help cells grow are permanently turned on, meaning that cells, they permanently grow and grow and grow, which can change more tumor suppressor genes. Now we got cancer. Now we have those cells that grow and grow and grow, make more and more copies, and they're all damaged cells because they've experienced a series of changes in DNA at specific sections of the DNA.